So this is the first half of the session 2, which is the research snapshots. So in this session, we will have two speakers, Jack Jameson and Charlotte Morgan, and they will be speaking, followed by some questions. After this, we will have a short tea and coffee outside. You can bring that tea and coffee back inside, which if you want, and then after that, we'll be coming back for the second half of the research snapshots. So to start then, we have Jack Jameson.
Okay, thanks Deirdre. So for part of my PhD, I've been looking at the performance of plough, mintill and direct drill systems, and to that end we had three main research objectives. The focus of today's presentation is the first objective, and that was to try and compare the on-farm and control trial performance of these crop establishment systems. The other objectives were to examine the economic performance of these systems and to try and model the soil carbon change in these systems over time. So to address the objectives, we had two main studies. So we had the Knockbeg trial and an on-farm study. So taking the Knockbeg trial first, Dermot spoke about it earlier. So it's a large long-term experiment looking at the impact of system and rotation in combination. And within that larger trial, I was monitoring crops of first wheat, followed after winter oilseed rape. So in this crop of first wheat, the input levels and management for each system were identical, and we had 4 replications of each system or treatment. And this layout or design allowed us to directly compare the performance of each of the systems. And then looking at the on-farm study, so we had 21 farms in this study, 7 each of plough, mintill and direct drill. Again, similar to Knockbeg, we were looking at crops of first wheat. However, there was large variations in both the soils, the input levels and the management from farm to farm. And these variations meant that we couldn't directly compare the performance of the systems in this study, but the on-farm study was invaluable in terms of seeing the range of both performances and management on farms, and both of these studies were over the last 3 growing seasons. Okay, so to look at some results now, so taking establishment counts first. So in Knockbeg, all 3 systems were sown on the same date in the middle of October, and at the same seeding rate. So when the management was the same, what we seen was direct drill establishment counts were significantly lower in 2 out of the 3 study seasons. However, all 3 systems, as we can see, did achieve adequate establishment counts to support yield potential, which was very encouraging. Now, to compare that with what we seen on farms, so just to explain about what we're seeing here, so I suppose every triangle, square and circle represents an establishment count from a farm in one of the 3 study seasons, and then the black diamonds here represents the average for each of the systems. So on farms there was high variability in establishment counts on the farms, but as we can see, most of the farms achieved very similar and adequate establishment counts to support yield potential. Now the difference with what we've seen in Knockbeg is that direct drill doesn't seem to have lower establishment counts than the other systems. Now this is mainly because direct drill growers were sowing 2 to 3 weeks earlier and were using higher seeding rates to try and, I suppose, counteract the poor establishment or the risk of poor establishment we've seen in Knockbeg. So now we're going to have a quick look at some growth data, taking Knockbeg first. So what we measured was light interception. Now light interception is measured because the more light a crop is intercepting, I suppose the more growth that has occurred across the season. And we've growth stages across the bottom here. So what we found in Knockbeg was that direct drill, I suppose, suffered from slower early season growth, but generally speaking caught up by growth stage 37 or flag leaf emergence. To compare that with what we've seen on farms, I suppose on farms growth was very variable within systems but comparable across systems. So as we can see, there's no great difference amongst the systems from what we can see here. The difference with what we've seen in Knockbeg is that direct drill didn't appear to suffer from slower early season growth. Now this is probably because with the sowing 2 to 3 weeks earlier and with the higher seeding rates, larger canopies were being carried into the winter, meaning there was less ground to be made up come springtime here in growth stage 21 and growth stage 30. Now yields, all important and interesting. So I suppose in Knockbeg, which was very, very encouraging, there was no statistical difference in yield across the 3 seasons, and I suppose you could see very good yields were achieved. On farms, things were a little bit different, so I suppose before we get into it, all the fields in the study years achieved very reasonable yields, but as we can see, direct drill yields or direct drill sites did seem to yield that little bit lower. However, this was mainly down to management, we think. So direct drill growers, in this study anyway, were using significantly lower levels of inputs. Now this was embodied by, on average, a use of 20% less chemical nitrogen. So these growers are purposely choosing to use lower levels of input. So it'll be interesting once the economic analysis comes around to see, does this reduced spend on inputs compensate for some of the yield depression we're seeing. In terms of key messages to conclude, so on-farm studies versus research trials. On-farm studies, so they were very interesting, very, very useful to see the range of both performances and management on farms. However, it was very challenging to compare the systems in this way due to the variation we've seen from farm to farm. And then in terms of knockbeg or the replicated research trials, so they are limited in the number of factors we can look at in them, but they're still essential to compare the performance of these systems. And then in terms of the establishment systems themselves, so as we've seen in knockbeg, which was very encouraging, these systems were all capable of producing similar yields. However, with an eye on long-term sustainability, we need to be mindful of the challenges of both grass weeds and, I think, wetter autumns. And I know the non-plough systems are probably more challenged by these 2 factors, and whilst they do bring certain benefits with work rates, etc., we do need to be conscious that ploughing will be a very, very important tool to have in our toolbox going forward. So I'd like to thank all the growers for participating, of which some I see here, and everyone else who were involved. So thank you.
Thank you, Jack. That was great. So next we have Charlotte Morgan. And just before Charlotte starts there's a car with the lights left on. The reg is 202C5409, so if that's yours, if you'd like to check on it. So go ahead, Charlotte.
Great, thanks a million, Deirdre. So why can poppies become such a problematic weed? Well, firstly, they're both autumn and spring germinating. They are a very competitive plant, and from a resistance perspective, they're actually the 3rd most important weed in Europe, right after blackgrass and Italian ryegrass. And this is due to the fact that they are cross-pollinating, they create high seed bank populations, and they also have a longevity of what's estimated to be greater than 10 years. And finally, they can evolve both target site and non-target site resistant mechanisms, which I'll talk about in a couple of minutes. So what are our herbicide options for controlling poppies? Well, some of these are just examples, but what I really want to focus on are the modes of action that are available for us. So firstly, you have your ALS inhibiting herbicides. So the likes of Camiomax, Alimax, and Pacifica Plus, these are your SUs. And then you have a slightly different chemistry in Broadway Star, but it is still an ALS inhibitor. Then available to us are hormones or synthetic auxins, so the likes of Pixaro with that newer chemistry Aralex in there. And you can also have a combination of both of these, so a hormone and an ALS inhibitor in one, which is something that we see in the likes of Zypar. Now these are all predominantly used in cereal crops, so for non-cereal crops such as winter oilseed rape, we have the options of something such as Astrocurb. So before the 2022 harvest, we received 2 suspect-resistant populations of poppies, one from Meath and one from Kildare. When we received these seeds, they were grown in a glasshouse, and they were sprayed with herbicides at the 4 to 6 leaf stage. So this is pretty optimal conditions. Now the results I'll be showing you will be from 4 different ALS herbicide trials. And when we sprayed these herbicides, we actually sprayed them at a variety of doses, from just a quarter of the recommended field rate all the way up to 8 times the recommended field rate. And this range really allows us to examine the extent of resistance in both populations. So the next couple of photos that I'm going to show all follow the same format. So our 2 rows are our 2 populations, the top one being from Meath and the bottom from Kildare. And as we move from left to right, we go from an untreated, unsprayed control plant all the way up through the doses to 8 times the recommended field rate. And that field rate is indicated there by that red 1X. Now this photo was taken looking down on the poppies in the trays in which they were grown and sprayed. So if we look at the effects of Broadway star first, we can see it's not until about 1.5 to 2 times the recommended field rate until we start to see control of the two populations. Now that's obviously above the legal limit that we're allowed to use in field, so if we focus in on that field rate, we can see that there is still growth in both populations, and this indicates some sort of tolerance or resistance. Now if we look at Ali Max and the effects on our two populations, we can see that even up to 8 times the recommended field rate, there's very, very little control, and there's still strong growing plants in both populations. And we can say the same for Cameo Max. At 8x the recommended field rate, still very strong growing plants. And finally, we saw the same again with Pacifica Plus. These 3 herbicides were virtually ineffective on both of these populations. And what I wanted to know is, why does this happen? So I started to look at target site resistance. Now, I'm not going to bore you with too much of the genetics of it all, but when a herbicide is sprayed on a sensitive plant, it can find its target in the plant and bind effectively, and this is what results in plant death. Now, how we look at this is, we look at the gene that creates the target. So with ALS inhibitors, we looked at the ALS gene, and this is something that we saw in all of our sensitive poppies, this sort of code here. Now, when a plant evolves target site resistance, that target has changed its structure so much that the herbicide can't bind to it anymore. And this is why we see strong growing resistant plants. And in all of the plants we looked at in both populations, we found a mutation in the ALS gene. And at this position, this mutation actually infers resistance to ALS herbicides. So this explains exactly what we just saw in the glasshouse. So what can we take from this? Well, firstly, these two populations have evolved target site resistance to ALS herbicides. And these two populations are actually the first cases of herbicide resistance that have been identified and verified molecularly in Ireland. Presumably, there are a few more out there. So we need to be vigilant with our weeds and herbicide efficacy, and resistance testing is critical. In the field, we can't rely solely on ALS herbicides. So using alternative modes of action is crucial. So if our target weeds are poppies, using a hormone-type herbicide can be very effective. And finally, integrated weed management approaches. These are crucial in the field, such as cultural control methods. They can really help to alleviate weed pressure. And on that note, I'd like to say thank you to everyone involved in the project. Just a quick reminder that if you do think you have a problem with resistance in your field, we do offer free herbicide resistance testing in Oak Park. And you can find out more about that from the workshop later on, the stand outside, online, or from your advisors. Thanks for listening.
Okay, thanks a million, Charlotte. That was great. Okay, so we have time just for a few questions then. So just to start then with yourself, Jack. In Knockbeg, why did the direct drill system have slower early season growth compared to the other two systems?
There's a couple of different reasons, but the main ones relate to the start the crop gets. So if you look at a plough-based system or min-till system, there's generally better soil-to-seed contact, better seedbed drainage, less smearing of the soil, probably more nitrogen mineralization, which gives the crop a little kick at the start. And as well as that, tilled soils generally warm up a little bit quicker in the spring, allowing growth to take off a little bit earlier. So they'd be the main ones, I think.
Okay. I suppose this here on slide then, like yield difference between direct drilling and other systems are interesting, but margin is what is about. What can we expect to see from the economic analysis?
I'm in the middle of doing it at the moment, so I'm hoping in the next 2 or 3 months we'll have some raw figures. So yeah, Shay and Dermot are giving me a handout, but there's a lot to it now when you get into it. So we're hoping that we'll have some results by the end of the summer, maybe.
Okay. Then yourself, Charlotte. You mentioned poppies are cross-pollinating. What does this mean in the context of resistance?
Yeah, so when a plant's cross-pollinating, you know, all the information for the resistance is in a plant's genes itself. So if it can create new seeds in a population, that resistance can spread quicker and further, and it can create more complex resistance within different plants.
Okay. I suppose, and like with such reliance on post-emergence contact herbicides to manage the poppy, should we be embracing pre-emergence more, do you think?
Yeah, it's a good question. I suppose it depends on the population, but I know BASF, they do recommend that pendimethalin herbicides can be used to control poppies. So the likes of Stomp Aqua and those sort of products, yeah.
Okay. And back to yourself then, Jack. Why not reflect on farm reality when the knockbag trial by direct drilling earlier? I'm not reading that wrong.
This wasn't a simple one, and I think Dermot was before my time, but there was a lot of thought put into this. But basically, a lot of the IPM advice around grass weeds, disease pressure, aphids are, I suppose, saying the opposite than sown earlier. So they're actually, from an IPM perspective, is to sow later. So we stuck with what the advice around IPM was and decided to sow on the same day. And I suppose when you're keeping the management the same and sowing on the same day, it's easier to compare. When you start saying we're doing this for direct drill and not for plough and vice versa, it all gets very messy and very difficult to compare the systems that way, you know.
I know you're saying you're only kind of starting on the economic analysis, but when it comes to the economic preferences, what key indicators will be considered, and any specific practices or systems that do you think stand out, particularly beneficial or challenging for farmers?
Okay, so the on-farm study over the last 3 years, those 21 farmers, they will have provided me with all field operation data, herbicides, fungicides, pesticides, fertilizer inputs. So all of those are going to go into the analysis, and we're going to do like a standard costing type approach. So we'll be given a cost to a kilo of nitrogen, phosphorus, potassium, and all the crop protection products. So we'll see then what the margin comes out at the end. Everything is being considered.
Okay, great. Then for yourself, Charlotte, when you say poppies are competitive, how competitive are they in relation to yield loss?
Yeah, so if you, I think judging that by weed competitive indices, so I know these have been in a few Teagasc crop reports before, but looking at poppies, they have a competitive index of about 12.5 plants per metre squared, and that can result in a 5% yield loss. So to put that in context, that's the same number as blackgrass has, and it's actually twice as competitive then as chickweed again.
So huge, can be a huge issue. Yeah, okay. Then does knowing the site of the mutation speed up the identification of resistance weeds?
Definitely, yeah. So different mutations at different positions when they're identified either with your ALS herbicides or ACK, so FOP, stims and dens, you can then, if you find that mutation, you know, okay, this isn't going to work in it and this isn't going to work. So and cut out a lot of the tough middle work for a lot of people, trialing different things before you realise it's not worth it anymore, it's not going to work.
Okay, and it's just one more question after coming in for you here as well. When spraying poppies in the glasshouse, how small are these weeds, and how does this mirror what happens on the farm? Or I know it's a difficult kind of a thing to compare.
No, it is a good question. So I think we do see, we try to spray it at the 4 to 6 leaf stage, which is often recommended on a lot of the herbicide labels that we use. So we try and stick to as much as what we could do in the field as possible and what's recommended.
And then, as you were saying then in your presentation, people can send in samples if they think they have a resistance issue. How long normally would it take to get back the results from them?
Yeah, it's also a good question. So it does differ depending on the weed that's emitted. So things such as blackgrass, this has a dormancy. So that dormancy takes 6 weeks to break before we can even start testing them. And then there can be issues with germination or not and resources. So not a definite answer, but ideally 6 to 12 weeks latest, hopefully, if all going well.
Okay. Then yourself, Jack, you said that direct drill growers are using lower levels of inputs in general. Why do you think this is?
From talking to the guys, it's not that direct drilling allows you to use less inputs or nitrogen or anything like that. It's a conscious decision from the direct drill growers in this study anyway to try and target a moderate or low input system, and direct drilling just happens to be part of it, because it's a lower cost establishment system. So yeah, it's just a by the way, I suppose.
So you're going to take that into consideration then, obviously, with the economic analysis. Okay. Then again, for yourself, how flexible were either established group about sticking or changing to their system with conditions in mind?
I think for the most part, most of them were not that flexible in that. I think the advice Teagasc have been given out for years is that you can't justify having 2 or 3 systems, so a lot of them would pick their system and they tried to stick to it. Now, having said that, some of the min-till growers we have in the study, they did keep the plough, and if needed, they have no problem in using it, which is probably a good pragmatic way of looking at it.
I'll just take one more question then for yourself, Charlotte. We're just out of time here now. So how big of a risk are we importing, or are we by importing resistant poppy problem with wildflower mixes?
That's a good question. It's something we haven't exactly looked at, but you know, when they are in these wildflower mixes, cross-pollinating, they can probably spread further, but it's unknown if they're coming in resistant or things like that. So because of those high seed bank populations, it is possible that they do stay in the seed bank for a long time. So it could be a risk.
Look, thank you very much. We're just out of time here now. So thank you to Jack and Charlotte for their presentations and answering questions there. So, okay, everyone. So welcome back there after coffee. So this is the second half of the research snapshots section. So this time we have Dr. Stephen Kildea going to be talking about barley seed health, and Dr. Attica Rahman discussing building opportunities for oats. So first up is Stephen.
Okay. So thank you very much, Deirdre. So as my title says, I'm going to give a bit of an overview of some of the research that we have done and some of the research that we are currently doing from a barley disease perspective. I'm presenting on behalf of Rabiza Zia. She's a PhD student. She's currently in France on placement at the moment, so I'm presenting some of her work on her behalf. So just to get straight into it, I suppose, I'm specifically looking at some of the key foliar diseases. I'm not talking about our traditional, I suppose, smuts and bunts, etc., but thinking of the foliar diseases that might be in barley crops. And I suppose the key ones would be Ramularia, Rhynchosporium, net blotch, rust and mildew, and nodorum. But some of actually these diseases potentially can be seed-borne. And I'm particularly talking about Ramularia, Rhynchosporium, and net blotch. So if they're seed-borne, is there a potential there that I suppose they're in Irish stocks to start with? And then I suppose the question then might lead to what implications that might have. And I suppose looking at the Ramularia specifically, this is some work that we would have carried out back a number of years ago as part of the Department of Agriculture funded project EPIC. And in that we used a molecular approach, qPCR, to actually screen seed stocks from an Irish perspective. We were fortunate enough in the 3 years of the study, this is going back a few years now, to get a sample of everything that would have been submitted to the Department of Agriculture for certification. So we're talking up on close to 700 samples are included in this. We looked at it from a Ramularia perspective, and with the qPCR, we specifically looked at either is it present or is it absent. We can look at it from a quantitative, but as I'll get into in a few moments, that can be a little bit more difficult. So from purely a qualitative presence-absence, what we could see is that there was a certain amount of the crops every year, or seed I should say every year, had a bit of, a good proportion of Ramularia was present. It did slightly increase in the years of the study, going from 2015 to harvest to 2015 to the harvest to 2017, but not really significant. What we did see was a significant difference between whether it was a winter crop or whether it was a spring crop, with a higher proportion of the spring crops having detectable levels of Ramularia in it. We also looked at where that seed was coming from, and remembering that we were looking at the multiplier here, not necessarily the field location, but where that sample was coming from the multiplier. And we didn't see a significant difference between where in the country that was coming from. So there was quite a good proportion of the crops have Ramularia in it. But as I said, that was very much a qualitative. We were limited in could we actually determine what level was in the seed based on the assay, because within the quantitative PCR assay, it is very much dependent on creating standard curves. And of course, because we're working with a seed here that has a huge amount of carbohydrates in it, it can impact the quality of the result that comes out of it. So we have looked at actually, can we bring that further and develop a method where not only can it be qualitative, it can also be quantitative. And that's where we've looked at a method called digital PCR. So the concept is still the same. We get our seed sample, we actually get the DNA from that seed sample, but within the assay itself, within the method itself, it is a bit more robust and isn't as impacted from sort of the carbohydrates or some other parts or quality of the sample that might be there. So with this, we actually said, look, we look at the Ramularia, but we know there's other, as I said, diseases, key foliar diseases that potentially could be in the seed. So we expanded it and said we'd look at Ramularia, but we'd also look at Rhynchosporium and net blotch. But interestingly, with the net blotch, we know there can be different species. There can be the spot form or there can be the net form. So we've invested in looking and said, okay, can we distinguish between both of those? And within that, we can. And I suppose what I'm only presenting here is a very quick look at what the data is looking like from that. But if we look for the net form, and in the winter crop, these are winter samples from about 2015, we could see those above the blue dot. Where there is a dot, it means that sample is positive. So we could see that quite a few of the samples that we had in this run were actually positive, but they had a relatively small number of positive dots. So quantitatively, it was quite low in that batch of seed. And that's the net form that we would have been looking at this. But interestingly, when we looked at these samples from a spot form, we could also see that above that green line, there's also quite a lot that are also showing positive. So looking at the sample, we could actually go back and look and see, well, what sample is this? And within these samples, we could see that that was the variety tower. And actually around this time, about 16, 17, and 18, the variety tower was developing quite a lot of the spot form of disease. So we know that actually method we can, I suppose, start off, we can detect these species within the seed. We can now also quantify the level that is potentially there with using this method in a lot more confidence than we previously could have. We've done the exact same for Ramularia, and of course the positive samples that we saw for Ramularia previously with the qPCR are positive. But because we've gone that little bit more sensitive, we can see that a lot more of the samples that we would have regarded qualitatively in the previous assay as negative are identifying with a very, very low level of Ramularia also. Equally with Rhynchosporium, we have the same run or same reactions for Rhynchosporium, and we can see some samples identifying with Rhynchosporium. So it is that the method that I suppose that we have been looking at and developing, we can utilize it going forward. And I suppose from a research perspective, it's very important for us to be able to detect this in the seed. And from Rebeza's perspective of her PhD, she wants to understand, I suppose, the Ramularia, how it develops in the crop, and going from the very, very start, from the seed through to the crop is really important. But I think from an aspect of actually understanding disease control, we have to remember that seed is only one source of inoculum. And this is something that we will get into in a little bit more detail in the afternoon in the workshop. But really understanding, I suppose, the susceptibility of the variety. And it gets back maybe to that previous chart where we could see the spot form in that blotch. We could see that that was related very, very closely to the variety that was specifically there. I should say when we run spring barley samples, we found no spot form in net blotch. We find the net form. Again, understanding the variety and relationship with the variety can give us that information too. But really understand, okay, then what are the other components of a disease management in spring barley or barley? It's basically going to be, okay, where is that crop going to be sown? And as I say, this will be something that we will get into in the afternoon. But understanding, trying to prevent and suppress that disease, it is very important. And it is, I suppose, the first part of IPM. And going forward, we will try to invest in terms of, okay, what is the implications of having these diseases in the seed? So that's really just a very quick sort of summary in terms of the research that we've been doing from a seed health and those diseases that I suppose aren't traditionally maybe regarded as seed diseases. But finally, I just want to maybe bring your attention to some work that we are doing, and I know it's moving away from the barley specific perspective, but we, over the next couple of weeks, are hoping to maybe send out a survey to some of you in terms of relation what the impacts of some of the pesticide reductions that might be coming down the line. We want to get a handle or an idea on what impact that might have, and we will be approaching you, I suppose, over the next couple of weeks with a survey. If you do get it, please respond very quickly because it is something that we do want to address. So with that, Deirdre, I'll pass it over to the next speaker.
Great. Thanks a million, Stephen. So next up then we have Dr. Teagasc Raman. So just also to remind people, if you have any questions for our speakers, please just put them up on Slido. Okay, so go ahead.
Thank you, Deirdre. Okay, so I'm not going to get into details of all of the research what we are doing, rather it's a snapshot presentation, so I'm going to give an overview of what major projects what we are undertaking currently. So yes, benefits of oats are real and proven. Oat has dietary fiber which helps to lower blood cholesterol and glucose level. Naturally, it's a whole grain and gluten-free. Also, oat as a feed has similar value to barley and maize. On top of that, in terms of sustainability, oat is low nutrient required crop, also has low carbon footprint, and also oat as a bread crop is as good as oilseed rape and beans. Now, Ireland has a rich history of growing oats more than 1,500 years. However, since 1916 to recent years, oat has lost more than 90% of its lands. In the meantime, its average yield also increased more than 63%. And good news is Ireland is one of the high-yielding countries when it comes to growing oats. However, that doesn't mean we don't have challenges. There are lots of other challenges, but we are going to focus some of the prime challenges or the most important ones in our current projects, and we'll be addressing more in our future projects as well. One of that is Teagasc commitment to develop and deliver the winter oat guides. What do you expect to see in this guidebook? So we will have answer for questions such as which component of yield, for example, grains per square meter, grain per penicle, or penicle per square meter, which of these contribute to final yield contribution. Besides the yield component and yield, there will be lots of information regarding growth and development pattern, such as these two graphs, which is showing that how shoots per square meter and green area index are changing over the growing season. One good news for you is that we have completed 2 years of data recording, and we have taken more than 3,500 data points. And 1 year you have to wait, but at our next year National Tillage Conference, we will have this guidebook delivered to you. We also looked into the nitrogen response, and our previous nitrogen response curve was developed based on the variety Bara. And now we included two relatively new cultivars such as Husky and Isabel. What did we find? We found that Husky and Isabel has significantly higher yield potential compared to Bara. But the interesting part is they don't demand more nitrogen than what was already recommended. So that is an indication that Husky and Isabel, which are relatively new varieties, have better nitrogen utilization and uptake efficiency. And that indicates that's a good sign that we don't need to increase our nitrogen recommendation. Another interesting project which is going on in between Teagasc Oak Park and Moore Park is the nutritional profiling of oats. Now, for this project, we have evaluated a panel of 574 oat varieties, which includes some heritage lines released as early as 1850s and also land races. And based on their agronomic and grain physical characteristic data, we have narrowed down to 90 varieties. These 90 varieties was analyzed for their different nutritions, and what we have found is really amazing. We are seeing that heritage lines have rich diversity when it comes for nutritional composition. As you can see, for example, the beta-glucan is ranging between 2.8 to 6.7%, whereas protein is reaching around 22%. So for our subsequent work, we also selected 5 varieties, and they were compared against the standard variety Husky. So Husky was given a scoring of 100% for nutrition and agronomic score. And as you can see, all of these selected varieties have higher nutritional score than the Husky and comparatively acceptable agronomic scoring. Especially the variety Difed, you can see it's more than double the nutritional when it comes to the nutritional composition against Husky. As we expect that oat production area will increase in Ireland, so does we are also afraid, or we are cautious that there could be outbreak of different diseases. One of that devastating disease is the oat mosaic virus, or OMV. For that, we aim to develop a rapid OMV detection method, which will help us to quantify the OMV, and also that will give us an opportunity to identify the lands which are infected with OMV and also the prevalence of OMV in Irish tillage sector. So it was delayed due to some technical issues, but it's going to start this year, and we will start this with the soil sampling this summer. But for that, I need your help. I need your information to identify some suspected OMV fields. That field doesn't need to be under oat cultivation right at the moment, as we will collect the soil, and if you have any information which could be previously known that that field was infected with OMV or suspected of infected of OMV, please let me know, get in touch with me. With that, I want to thank all of our collaborators and funding agencies, and also especially all of you for listening. Thank you so much.
Thanks, Adik. Okay, so now we have time for some questions. Okay, so we'll just start with yourself, Stephen. Does presence of ramularia in the seed impact control later on in the season?
A very good question, Deirdre.
I suppose that is something that we don't have the full information on. We know that it can move from the seed into the crop, but I suppose what really affects ramularia then is going to be the environmental conditions that come after it. It is something definitely we are looking at to see, look, if you have a high level of disease in your seed lot, will that mean that you will have a high level later on? But it is something that we don't have the exact answer on yet. But definitely there is the research to demonstrate that it can move from the seed or into the plant.
Okay, and then I suppose kind of following on from that then, what do you do if you suspect that your seed is already infected with ramularia?
Again, I suppose when we're looking at it from an aspect of the crop growth itself, ramularia is going to be a late season disease. So its implications will be, if it does have an effect, it will be late season, and it will be a combination of the environmental conditions that will prevail. So what we would be saying is that, look, it's really got to do with those from a disease control perspective. I think going forward, if we can find out that actually, yes, crops that are seed lots that have a lower level will lead to a lower level in the crop subsequently, then there potentially is measures to control it in that sort of aspect. But at the moment, from a control perspective, it is managing your crop as if it has ramularia.
Okay, great. And then I suppose just one more question then for yourself, kind of following on for that again then. What value would you place on disease-free seed compared to the samples in the survey that had Ringo, net blotch and ramularia? And when might we have a value?
Sorry, you'll have to repeat that question. So when we'd have, I suppose, the aspect of, look, if we know that a crop has a high level of the other two diseases, definitely, I think they can have an impact, I suppose, earlier in the crop life in terms of tiller and the shoot numbers and things like that. Definitely, if there's a very, very high level there, then actually some measures need to be taken. From ramularia, it is, as I say, a little bit more difficult because the implications are later in the season.
Okay, great, thanks. Then for yourself, Attic, you've mentioned about 5 selected oat varieties there in the Farm to Fork project. What is the next step in this project?
So, hello. Okay, sorry. Sorry. Yeah. The next step is we're going to put them in the field again, and we're going to study them for their nitrogen requirement and input requirement to adjust their agronomics. And also in terms of those 5 selected varieties, we will, in Moore Park, they're going to study how the different nutritional composition is being digested in our gut system and how that nutrition is being uptaken in human system. So yeah, that will be the next step.
And just then, of these varieties, can they be grown as a commercial crop, these heritage varieties?
So some farmers, they're small farmers, they're growing those heritage lines that I am aware of, but I don't see any problem of growing them. But before we recommend them to farmers, we're going to actually study their agronomic requirements, and we're going to tailor the agronomics for those specific varieties, and that's what we're going to do this year and the subsequent years, and then we'll have a concrete answer whether that can be really recommended to farmers or not.
Okay, yeah, so I suppose then it's kind of hard then to kind of describe whether they need drastically different management practices, I suppose, compared to the standard varieties.
So the results which I have shown, or the findings based on our results or experiment, that we didn't put any differential management practices for our heritage lines. So there all of those varieties, including standard checks like Husky, Isabel, and 2 more varieties from Aberystwyth University in Wales, they were under the same nutrition, same management conditions. So based on that, we actually selected these 5 varieties.
Okay. Just another question for yourself then. So when it comes to the oat mosaic virus detection method, when do you expect to have this developed, and when will it be available to the public?
So we hope to start the soil sampling this year, summer, and we already started working on the molecular detection method. And we expect to deliver the final detection and quantitative method in our next year Teagasc conference.
Just, I suppose, the top question there, it's got a good few likes. Why are more oats not recommended? Why are oats not recommended more in animal feed rations?
That's a good question, but difficult to answer in the sense we know that oat is good for as I mentioned, that, and also that's based on Teagasc and UCD study, that it's a similar animal feed value compared to barley and maize, but exactly I don't know why it's not being recommended. Probably from animal sector they can answer this question. But my suggestion would be to definitely include them in the feed ration. Yeah.
Okay. Then yourself, Stephen. The top question here: if a farmer gets certified seed tested and it shows up to have disease problems, would this lead to big problems for the seed trade going forward?
I don't think it would lead to big problems. I think it's actually an opportunity to understand what is in the seed. I think we know that these diseases will inevitably be there because that is part of their cycle. Understanding that what levels might be there, can we tailor it to limit that, be that a seed treatment, be it a case where there's a very high level in the seed, then it may be excluded. But I think actually understanding and knowing that is more an opportunity than a problem.
Okay. And then do you think there's a possibility of introducing more fungicide-resistant strains from imported seed?
Absolutely, no doubt. If the pathogen is there, it has what traits are going to be in it. Now, what I would say from a Ramularia perspective, look, we are probably, unfortunately, the point where actually there's a lot of resistance in our population already, so it's unlikely that we're going to introduce a new level of resistance. So we have to look at it that way. But definitely, look, what we grow in terms of the crop, if it has the pathogen, it's going to include those traits.
I suppose the top question then again there, would some of the biocontrol or biostimulants, do you think they have a role in controlling seed-borne diseases developing into the canopy?
If they have an ability to control the pathogen, absolutely.
Grant. Then back to yourself then, Attic. I think it's this for you anyway, will the variety DIFED be trialled for recommendation as a variety if it's superior for nutrition and agronomic characteristics?
So first we will have one trial in our Oak Park field, definitely, to understand their agronomic needs. But whether it will go to the recommended list or not, that's probably a long shot still. So, but yeah, definitely we have a big hope with this variety, and we will definitely give trial in our field. Yeah.
And then, I suppose, how do you think oat consumption has changed in the Irish population? Is there more opportunity for a higher value food grade that might encourage more growers to grow the premium crop?
Yes, I do think so, because if we look into different all the Scandinavian countries, and they are extracting multiple things from the oats. They are using oats and oat-based product for cosmetics, to medicine, and lots of other things. So definitely there is a chance of growing high-value food grade or similar to besides food, some other value-added addition of oats. And definitely there is a chance, yeah.
And then finally, Stephen, I'll give you the last question here. Is seed or environmental source of Ramularia different, or is one worse than the other, do you think?
I don't have the perfect answer for that. I think both are important, I'd imagine. Being honest, where that inoculum is coming from, it's still inoculum. So if it's coming from the seed, it's going to be important. If it's environmental, it'll be important too. So I think both are probably equally important.
Okay, that's great. Look, thank you very much. That's the end of this session then. So just thanks to Stephen and Attic for their presentations and answering the questions.